All living things are made of proteins, so it is important to understand how protein interacts with other proteins, drugs, liquids, and more. If you've ever made a stick and ball model of H2 or stage 4, then you've done scientific modeling. But if we put organic molecules together, specifically amino acids, you can think of them as the building blocks of proteins. Then we end up creating a protein model. Protein modeling can help us predict interactions that would otherwise take many years to understand. Without protein modeling, it is very hard to predict the way a drug interacts with a protein without going through an extensive and expensive guess and test process. Since proteins are rarely just a few amino acids, and some being hundreds to thousands of amino acids, this makes it difficult to predict their structure by hand. Thus, protein modeling is done using software that uses known similar structures to efficiently predict what a protein looks like. This software is usually developed by both computational, biological, and chemical researchers, and requires high computational power to run because under the hood, this software is performing hundreds of mathematical equations to give us the most likely structure of a protein. At iGEM, our projects always revolve around a protein of interest, so protein modeling is crucial to get a closer look and understanding of what our protein is doing.